And on to topic number 3 of this snippet collection of lectures, for technical reasons, becoming the new version of for historical reasons, I'm afraid. So now you know how glycogen is being formed, how it is being degraded again so that glucose, when required, is being released and then can be metabolized accordingly. And then we come to the point where obviously it becomes necessary for the organism to synthesize glucose de novo, starting from actually pyruvate. And this is required because there are some cells which, as fuel, can only work with glucose. There are the erythrocytes, as an example. I've already mentioned the cells of the adrenal gland, and both can receive energy exclusively by glucose, and the same goes for all the brain cells. So, as the glucose is hardly storable in the brain cells, and like the breaking down of glycogen to release new glucose might take actually too long, then it is well possible and necessary that glucose has to be synthesized de novo, particularly if the food you take up or the organism takes up does not contain enough glucose, either in free form or by starch-containing foods in a form that's easily biodegradable within the organisms. Now, please do not make the mistake of assuming that trying to feed yourself with glucose exclusively would be a healthy lifestyle. It is not. Glucose is definitely important, but it's not the only thing the body needs. Some of the other things that are required for instance, the human organism will pop up in the lecture sooner or later on. But so, in a nutshell, you need glucose, but you definitely should not try to live exclusively on glucose. I know this sounds a little bit odd coming from somebody like me who seems to exclusively live off wine, gum, and gummy bears when in the office, but occasionally I eat something else as well, and apart from that, I'm not supposed to be your role model anyway, so in brief, don't. Okay, so the synthesis of glucose starting from pyruvate, it's not a pyrite, pyruvate, once again sounds as if it were just the backward reaction series from glycolysis, because you might remember that starting from glucose, you in the end had phosphoenol pyruvate, which was then hydrolyzed to form pyruvate. So it stands to reason that one makes the mistake of assuming the de novo synthesis of glucose is just the reversal of the mechanism of glycolysis, but it is not, because there are some steps which are simply irreversible. So we're not talking about an exact reversal of glycolysis, just like the degradation of glycogen is not the exact reversal of the synthesis of glycogen. For the same reasons, because some steps are actually not reversible. Now, in glycolysis, with which I hope by now you're a bit familiar, there are 3 steps from bottom to top. The phosphorylation of glucose to form glucose 6-phosphate is not reversible. Neither is the phosphorylation of fructose 6-phosphate to the fructose bis-phosphate, and the final hydrolysis of the high-energy intermediate product of phosphoenol pyruvate, our beloved PEP, to pyruvate, leading to the formation of ATP, which, as you will probably remember, was one of the reasons why the whole organism does the whole rigmarole of breaking down glucose to smaller C3 fragments from the C6 body glucose to 2 C3 bodies pyruvate, because ATP is being produced, and this, of course, is our energy currency number 1. So, the net result of the de novo synthesis, the gluconeogenesis, the de novo synthesis of glucose, is of course the reversal of this step, but it's just the net result, because actually there are different reactions involved. In many cases, different enzymes are involved. Occasionally, the enzymes actually can catalyze the forward and the backward reaction to the same extent, provided we're talking about a reversible reaction, not all of which are. Look at those here. Excuse me. And actually, since different reactions take place, different mechanisms are involved, occasionally you even have different intermediate products, as you will see starting with the next slide. Now, the gluconeogenesis, the formation of new glucose, occurs predominantly within liver cells and kidney cells. That does not mean that all the other cells are completely unable to do so, but the liver and the kidney cells are best equipped to do that. That's why this happens predominantly. Okay, there. Okay, so let's have a look at the first step. How can we start with pyruvate and in the end up with glucose? Now, you know that the glycolysis is in essence an exergonic reaction series. Let's not call it a reaction, because you know each of the intermediate products being formed are the products of an individual reaction, each of which being catalyzed by one or the other enzyme. And in the end, you know that due to the phosphoenol pyruvate basically being the final product of the glycolysis, with the last step, the hydrolysis of the phosphoenol pyruvate to pyruvate, is the reason why ATP is being formed. Well, if you look at that, then you already see some of these reaction steps here, like for instance, the hydrolysis of the PEP to pyruvate. Once again, and here we're at the topic of controlling reactions and control points of the reaction series, are controlled by one or the other substance. For instance, in the phosphoenol pyruvate, this is a typical example of a product-controlled reaction. If the level of free ATP in the cell is large enough, then the phosphoenol pyruvate is not being hydrolyzed any further, because then, of course, further ATP would be synthesized. And if you want to think of Le Chatelier once again in this case, please be my guest, because basically that's what's behind it. Since the concentration of the product in the solution, in the mixture, is large enough, there is the equilibrium, so to say. It's not a real one, because this is one of the irreversible steps, but the equilibrium, if you allow me to describe it like that, using quotation fingers, of course, lies on the reactants side, that is on the PEP, if ATP is already present in larger amounts. And the same goes for several other steps, like for instance the phosphorylation actually of glucose to glucose 6-phosphate also is controlled by the concentration of G6P. If there's already enough of it, then the phosphorylation of glucose to form G6P will be hindered. And the same goes for this third and final control point. I want to say something about that, which is the phosphorylation of the fructose 6-phosphate to FBP, which in the end, not in this step of course, but in the end will lead to the formation of ATP. So there is a control point at far higher up in the hierarchy, so to say. If there is enough of the end product, this end product already inhibits the further metabolization of the intermediate product fructose 6-phosphate. Okay. Now, the enzymes involved for each of these three control steps or controlled steps have already been discussed when we talked about glycolysis in general. So let's see how we can go and try and reverse that using different mechanisms. Please, once again, do not assume it's just the reversal of this reaction series, because it is not. Okay, there is one more step in our step from GAP, glyceraldehyde 3-phosphate, to the 1,3-bisphosphoglycerate, brackets, which contains two phosphate groups, no doubt, but they are not chemically identical. One of those is just the ester of the glyceraldehyde alcohol being esterified with the phosphoric acid, Whereas the phosphorylation of the glyceraldehyde side, so that the aldehyde function is being oxidized to form a carboxylic acid, which then forms a mixed anhydride with phosphoric acid, so that we end up with the 1,3-bis phosphoglycerate. Now, this obviously, there obviously is an oxidation step involved. If you look at the oxidation number of the carbon in the aldehyde and in the resulting carboxylic function then being part of the mixed anhydride is changed. The oxidation number is increased. So there is an oxidative material, excuse me, there is a reaction part present, which of course then is to be has to be reduced. So it would be NAD plus, which is reduced to NADH. But if it's reduced to NADH, then this stuff, which actually is supposed to function as an oxidative reagent, has to be recycled from the now reduced form, which of course can only function as a reductive agent and then be oxidized in the same step. So this is another situation where you say this is not simply reversible here, because if you wanted to reverse this, you would have to reduce the carboxylic function to the aldehyde function, and for that, of course, you need a reducing agent that is a reducing equivalent. Okay, now let's have a look. In the first step, our C3 body, the pyruvic, I dare say you're familiar with it by now, is carboxylated to form a C4 body. Well, oxaloacetate is a molecule you've seen, and be it only with the example of the aminotransferases I just mentioned something about in the last video session. But it's quite interesting that you want to convert a C3 body to a C6 body in the end. You want to go from pyruvic to glucose. Don't forget what we're talking about here. And the first thing is the formation of a C4 body, and that under using up energy. You use our ATP once again, which of course then is being hydrolyzed to form ADP, and the phosphate group is being split off. Well, let's have a closer look at how this reaction works, because once again, we're not talking about biochemistry is magic and somehow carbon dioxide, which of course is present in the cell, reacts with pyruvic and suddenly you've got oxaloacetate. There are several things you should keep in mind if you look at that reaction. For instance, a cell might and will contain carbon dioxide, no doubt, but that does not mean that this carbon dioxide really is present as the molecule CO2, which you know to be gaseous at room temperature. I know some people are really bloated figures, but that's for some other reasons than the presence of carbon dioxide, and I should stop talking about American presidents. Anyway, we should have a closer look at how this reaction takes place, because there is something happening here, which is actually one of the major things in organic chemistry, the formation of a new carbon-carbon bond. It is this carbon here reacting with the carbon of carbon dioxide to form this newly formed CC bond. These CO2 down here, that's our former carbon dioxide. So, how is this possible? And I hope it will at least alleviate your fears of biochemistry in general if I tell you that you already know all there is to know about the reaction mechanism here, because you did that in organic chemistry. Now let's have a closer look at this pyruvic again. You know, it is this carbon to form this bond here. So what do we see here? We are talking about a ketone. Yes, that one of the binding partners of the carbonyl carbon of this ketone is a carboxylic carbon, might make pyruvic a slightly odder substrate than you're used to from normal organic chemistry. But in essence, we're still just talking about a ketone with one substituent we're not interested in, and this methyl group. In other words, we've got a carbon bearing a hydrogen in alpha position to a carbonyl carbon. And you know that this means enolization can take place. The keto form with the CH3 here can also go into the enol form with the CC double bond here, and the hydroxy group add our formerly carbonyl carbon. Then, if we're talking about this enol, you know that any enolic carbon now has a higher negative charge density than you would normally expect it to have. In other words, it is a nucleophile, or a Lewis base if you prefer the inorganic term. On the other hand, the carbon in carbon dioxide is doubtlessly strongly positively polarized. Here carbon has the highest possible oxidation number, +4, and of course the two doubly bound oxygens deplete the electron density from this carbon at least to a noticeable amount, so maybe it's not depleting, but it lowers the electron density present on the carbon in a noticeable amount, leading to this carbon being strongly positively polarized, making it an electrophile, or a Lewis acid, depending on how you want to put it. Now, under the influence of the enzyme pyruvic carboxylase, and actually the presence of a cofactor, this time it's biotin, it's almost as important as our pyridoxal phosphate, you've heard something about in the last video sessions. And in the presence of magnesium 2+ ions, the role of which shall not be discussed here, but just for completeness' sake they should be mentioned, and particularly if acetyl coenzyme A, and you remember the coenzyme A from the energy transporters and the reductive equivalent, or redox equivalents and the like from the respective video session, if all of those are together here, then the formation of a new carbon-carbon bond is actually feasible. Well, it's driven by the energy being released due to the hydrolysis of an ATP, but by now it's just a normal nucleophilic attack of this negatively polarized enol carbon to the carboxylic carbon, to the strongly positively polarized carbon of our carbon dioxide. So, you should have some I remember moment here. Okay, this requires biotin. Just like I've shown the pyridoxal phosphate to you in the last video session, I think it was the last one, here let's have a look at biotin. This is, in essence, a carbon dioxide carrier. How that one works, you will find out in a few more slides. Now, this biotin is the prosthetic group of carboxylases in general. Well, at the moment, we use the pyruvic carboxylase. What a surprise, we want to bring carbon dioxide to pyruvate, so understandable it's the pyruvic carboxylase being used here, but there are other biochemical pathways where carboxylases also play a role, in which they also transfer a new carbon atom to an already existing CN body, which then becomes a CN+1 body. And it's always biotin doing this, so something must be interesting about this molecule. If you look at it, I don't know if you already have an idea where it might stem from. I'd be extremely surprised unless you happen to have learned that already in cell biology. And actually, at the moment, the biosynthesis of biotin isn't really important to us. All this down here isn't really that important. The most important part is this up here, which should remind you of urea. If these two nitrogen atoms did not have a nitrogen-carbon bond to the next binding partners, but instead were connected with another hydrogen each, then we'll be talking about the diamide of carbonic acid. H2N, C double bond O, NH2, that's urea. Okay, the nitrogens here will play the important role. The rest is not to be ignored, but the actual music plays up here, and you'll find out how that happens in a moment. For biotin, this is interesting. There are several names for biotin. Obviously, there's biotin. In the English literature, it's called vitamin B7. For reasons which once again go beyond the scope of this lecture, in France it's called vitamin B8. Don't ask. And in Germany, it's called vitamin H. Now that might be confusing, but actually this can be explained because when biotin was discovered to play an important role in biochemistry, it turns out that the lack, or at least a too low a value of biotin in the cells, led to changes predominantly in the skin and the hair. And since in German both skin, Haut, and hair, Haar, start with H, they said, well, this is obviously the vitamin for both the hair and the skin, so let's call it vitamin H. Now you know that. And I've just mentioned that this biotin plays an important role with carboxylases in several other biochemical pathways, some of which you will get to know during this term. For instance, in the metabolization of fatty acids, another topic we'll come to once we're finished talking about glucose in all its beauty. And let's see whether we'll come to the metabolization of various amino acids, but for some of those, not all of them, but some of those, once again, carboxylases are involved. So once again, the CO2 carrier biotin functioning as the prosthetic group of the respective enzymes are vitally important. Okay. So let's have a look how pyruvate is being carboxylated, one step after the other. We've got carbon dioxide within the cell, so only a minute amount of carbon dioxide will be present as carbon dioxide. You know that carbon dioxide is soluble in water. Not readily so. The concentration of dissolved carbon dioxide in water is relatively small. You know that, and you've noticed it the last time you opened a bottle of sparkling water, because the moment you opened the bottle, you heard the characteristic which was the sign of carbon dioxide evaporating or leaving the container. But the small amount of carbon dioxide being dissolved in water, will have reacted in water to form carbonic acid, H2CO3, which you know to be a weak acid, but acidically enough so that this H2CO3, this carbonic acid, will deprotonate, at least to a certain extent, in aqueous solution. That's why all the fizzy water tastes slightly sour, because the carbonic acid dissolved in the water will deprotonate, and as such increase the concentration of hydronium ions, so lowering the pH value. That's why it's called carbonic acid. Duh. And this carbonic acid now is once again converted into a mixed anhydride with a phosphate, so with a Pi, so to say. Now this Pi stems from ATP, so this phosphorylation of the carbonic acid, that is the anion of the carbonic acid, the hydrogen carbonate ion, to form this mixed anhydride requires energy. ADP is being split off, and then we will see what happens to that one later on. Now, by connecting this formerly hydrogen-bearing oxygen of the hydrogen carbonate with phosphate to end up with this mixed anhydride, this obviously doesn't change anything about the lack of electron density at this carboxylic carbon of our carbonic acid. In other words, this carbon is just as strong an electrophile as it was before. Now, there is something I've told you already in the first term. As long as you're not completely familiar with at least the most important elements in the periodic table with respect to biochemistry, which was CHNOPS, CHNOPS, you remember that, please always draw the lone pairs present. If you're versed enough in chemistry, you will give oxygen automatically to lone pairs, and one nitrogen, if it has got 3 bonds like here, it will have a lone pair. So you should remember to draw it there, but most biochemists don't do it because they're versed enough in chemistry already. So the nitrogen in our biotin bears a lone pair here, which, just like in the last video session, obviously is a center of high negative charge density. In other words, this nitrogen with a lone pair is a strong nucleophile or Lewis base. So this lone pair folds out to form a new bond with the carboxylic carbon here, which then of course would be pentavalent, which cannot happen. So this bond between in the mixed anhydride will lead to the release of a free phosphate. We can use that later on. And there is a new nitrogen-carbon bond being formed. Now, this prosthetic group of our biotin, this just means this is a non-protein part, which in this case is even covalently bound to a lysine rest somewhere within the primary structure of our carboxylase, in this case the pyruvic carboxylase. The enzyme in itself really is of no interest to us right now. You don't have to know the geometrical shape, the 3D shape. You don't even need the primary structure at the moment if you just believe me that this is the enzyme facilitating all the reactions here. So in other words, the prosthetic group, which just means is a non-proteinogenic part, in this case is connected to the epsilon amino group of one or the other lysine, and the rest of the enzyme really is of no interest right now. Okay, so at the moment we've got this I'm awfully sorry. So at the moment we've got this nucleophilic attack of the lone pair of the nitrogen to form a new nitrogen-carbon bond. The phosphate already has gone, and this intermediate, where this carbon dioxide, or this former carbon dioxide, which was present as the hydrogen carbonate ion in the aqueous solution, now explains why this biotin is called a CO2 carrier, because at the moment we've got a CO2 stuck to the nitrogen here, to one of the two nitrogens of our pyruvate, of our biotin. And now here we've got the enol form of our pyruvate. Remember, in the keto form, it just had the carbon-oxygen double bond here, and this hydrogen was part of the methyl group. Here is the enol form, in which the carbon is slightly negatively polarized due to this double bond here, so it becomes a nucleophile. So the pi part of the double bond is being folded out to this formerly carbon dioxide carbon. This, of course, would once again lead to a pentavalent carbon, which cannot happen, so the carbon-nitrogen bond we just formed is broken again, and this means our biotin is free to do the same thing to the next substrate, so the next phosphorylated hydrogen carbonate can also undergo this reaction, and the whole thing starts again and again. But the important thing is, by this nucleophilic attack of the enol carbon of our pyruvate, we convert this C3 body, 1, 2, 3, to a C4 body. So, suddenly, this all makes sense. Admittedly, the role of magnesium 2+ has not been discussed here, and why acetyl coenzyme A is of interest is something else we'll come to slightly later on. So, if you remember your glycolysis, you remember you had the C6 body to phosphorylated C6 body, G6P, to the conversion from the glucose to the fructose, so it was the isomerized phosphorylated C6 body. This phosphorylated C6 body, fructose 6-phosphate, was then phosphorylated a second time to the fructose bisphosphate, which then underwent the retroaldol reaction, so these C6 body, C6 body, C6 body, C6 body is being broken down to two C3 bodies, whereas the synthesis of glucose, the de novo synthesis of glucose, and that's what we're talking about here, goes via a completely different intermediate, a C4 body. Let's see what happens next. Now, this oxaloacetate, this C4 body, decarboxylates again. Now you might wonder why on earth biochemistry have made it that way, because it actually sounds a little bit like a waste of energy, doesn't it? Couldn't we have just phosphorylated our pyruvate to phosphoryl pyruvate? No, we cannot. This was one of the irreversible steps. We can only enforce the formation of the high energy form phosphoryl pyruvate if we hydrolyze, another nucleoside triphosphate. In this case, it's GTP, not our ubiquitous ATP. This is hydrolyzed, so one phosphate is being split off, and this happens. No, this happens is not correct. In the process of this phosphate group of the GTP being split off, this phosphate group is being transferred to the oxaloacetate, which decarboxylates at the same time, or something like that. Let's see what happens. So this is exactly what happens. The carbon dioxide is being split off again, and we end up with the phosphorylated form of the enol of a pyruvic again. Now this, of course, cannot return to the keto form, because here this oxygen already has two bonds, one to the carbon and one to the phosphate. This is just not as enolizable as, for instance, the hydroxy group of any aldose or ketos in the hemiacetal form can return to the open chain form if this hydroxy group is used to form a glycosidic bond or any other kind of bond. Once the hemiacetal or hemiketal are converted into an acetal or a ketal, then the reversion to the open chain form with the carbonyl function simply isn't possible anymore. Okay, so now we're stuck with the high energy molecule PEP, which is a good start. It's high energy, so sooner or later it will be able to release energy again. But first something else happens. First we've got this carbon-carbon bond to which water is being added, so that the hydrogen of the water ends at the carbon already being connected with the phosphorylated oxygen, and the OH group ends up here. Now, you might remember something like that from organic chemistry. If you return, mentally at least, to organic chemistry, the addition of water to PEP with the hydroxy group ending up here at carbon number 3 might appear counterintuitive because you've learned the Markovnikov rule. The Markovnikov rule in its simplest can be stated as if there are two carbons and water is being added to it, then the hydrogen will go to the carbon bearing more hydrogens already. So you would expect this CH2 to end up with a hydrogen becoming a methyl group, and this carbon here to get another hydroxy group, and then you'd have the hemiacetal with one of the two hydroxy groups being connected to an acid, in this case phosphoric acid. But this does not happen because this is an anti-Markovnikov addition. Please remember, it has got something to do with the question of which atom is polarized like which, what way, and how charge densities can be shifted. So, in brief, the addition of water to our phosphoenol pyruvic leading to 2-phosphoglycerate is an anti-Markovnikov addition, another word I've never used before, addition. Of course, it's catalyzed by an enzyme called an enolase. Since an enol is involved, this even makes sense. Now, an already known enzyme, the phosphoglycerate mutase, shifts the phosphate group from the secondary alcohol at carbon number 2 in 2-phosphoglycerate to the primary alcohol function at carbon number 3. So we end up with 3-phosphoglycerate. Yes, the phosphoglycerate mutase is the enzyme facilitating that, and then this 3-phosphoglycerate is phosphorylated once again using ATP, and you end up with exactly the same 1,3-bis phosphoglycerate, in brief BPG, you also know from glycolysis. So even though I've just told you several times that the reversal, so to say, reversal of the glycolysis, the de novo synthesis of glucose, the gluconeogenesis, is not the absolute reversal of all the steps, but some of those are extremely similar, whereas others obviously were completely different because there was no C4 body in glycolysis. Okay, so the phosphorylation of our 3PG to BPG leads once again, of course, requires an ATP because somebody has to deliver the phosphate. In this case, once again, it's ATP. And I've said it many times, I'll say it again until you're all blue in the face. Please remember that these two phosphate groups are not chemically identical. This is a phosphate ester of the alcohol glyceraldehyde, and this is the mixed anhydride of the carboxylic acid here, the oxidation, this carboxylic function here, which now forms a mixed anhydride, making BPG, of course, a high energy molecule again. Okay, now our bisphosphoglycerate is being reduced by NADH, which of course is the reducing form. So the oxidized form NAD plus is being produced here, and since NAD plus is an important oxidative reagent, actually it's quite helpful that this happens. And if this BPG is now being reduced, this leads to two things. A, of course, the phosphate rest at the oxygen of carbon number 1 is being split off. Now the GAP is only singly phosphorylated again as an ester of phosphoric acid, and the carboxylic carbon of this mixed anhydride is reduced to the aldehyde form. So here we are, glyceraldehyde 3-phosphate. The triose phosphate isomerase once again allows the isomerization of GAP to DHAP. So our aldehyde carbon present up here, actually this molecule should have been put upside down, then it would have even become easier. Because what happens is that the enolization of this carbon-oxygen double bond, and there is an alpha hydrogen here, would lead to the already known enediol, already known from the glycolysis, of course, and this enediol can either go back to the aldehyde form or it can continue enolizing, leading to the formation of a carbon-oxygen double bond here. So this carbon here is this carbon down here. And this, of course, is the same carbon as before, because this is the same fragment as this one. So the isomerization of glyceraldehyde triphosphate to dihydroxyacetone phosphate allows then these 2C3 bodies to undergo the aldol reaction leading to fructose bisphosphate. Once again, this aldol reaction is, of course, catalyzed by an enzyme, and since it's an aldol reaction taking place, it's called FBP aldolase. Now, I know this sounds very odd, but when we started with the glycolysis, I tried to explain to you how this the C6 body of FBP was broken down to 2C3 bodies via a retroaldol reaction. Now you could say the formation of FBP is the retroaldol reaction of glycolysis. So in other words, it's just an aldol reaction. And we're at FBP. And this, of course, is something you know from glycolysis. This can easily, FBP, this can easily be dephosphorylated to fructose 6-phosphate, then it isomerizes to glucose 6-phosphate, and there you are. That was the form of glucose present in an aqueous solution predominantly. The free glucose was rare, or was rarely there in a higher concentration. So let's compare once again glycolysis from glucose to pyruvate and gluconeogenesis from pyruvate to glucose. I really don't want to go into too much detail with all of the individual steps here, but some are quite important. So, for instance, for the synthesis of oxaloacetate from pyruvate, presence of acetyl coenzyme A obviously has a beneficial effect. So there we've got an allosteric activation of the pyruvate carboxylase. Why this is the case, you might find out later on, otherwise feel free to look it up in textbooks on biochemistry somewhere later on. We didn't really talk about acetyl coenzyme A at that length required at the moment, but you will encounter that one again and again, of course, because it also plays, for instance, a vital role in the citric acid cycle. Okay, then the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate actually is being hindered or deactivated by something quite similar but not identical, F 2,6-bisphosphate, fructose 2,6-bisphosphate, is a molecule we didn't encounter yet, but you will re-encounter it when we continue with metabolism pathways. But I think even though you've never seen it before, by now you should be able to even draw the structural formula of fructose 2,6-bisphosphate. You just have to shift the phosphate from hydroxy group of carbon number 1 to the hydroxy group of carbon number 2. And just as the presence of fructose 2,6-bisphosphate hinders the dephosphorylation of fructose 1,6-bisphosphate, the same presence increases the activity of the phosphofructokinase, leading to the formation of fructose 1,6-phosphate. So does the presence of AMP, whereas ATP once again counteracts or hinders the enzyme. Why is that the case? Because if we look at glycolysis again, the job of which, amongst other things, is the formation of ATP, the presence of ATP being the product, is another example for product control of a reaction step in an enzymatically catalyzed chemical reaction. Okay, the presence of citrate here might come as a surprise, but this is just because we didn't talk about the citric acid cycle. You obviously already did that in cell biology. I'm sorry. We could do that with each and every step. Some of those might be self-explanatory, like once again, the presence of ATP hinders the dephosphorylation of phosphorynyl pyruvic, because this reaction would lead to the formation of more ATP. That alanine plays a role here as a deactivating, as an allosteric inhibitor at the moment, once again, might come as a surprise, but this is about to change when we continue within this hopefully not too long video session. But no, for technical reasons, I'll have to cut this off again. The next video is coming up shortly.